This video is going to take us from pyruvate, which we ended up at the end of glycolysis, all the way to the rest of the CO2s. So we are going to completely finish oxidizing glucose here. And that is going to be primarily due to something called the Krebs cycle. So if we're talking about the oxidative breakdown of glucose, eventually to form ATP, we can think of this as being three separate stages. So the first stage is going to be up to the point of acetyl-CoA. Unfortunately, this is not exactly glycolysis. I'm going to call this one, which could sort of fit either way, the transition reaction. It doesn't have a good name, but some places call it the transition reaction. I think it's helpful to give it a name. And this is basically transitioning between the multi-step pathway of glycolysis and then the multi-step pathway of the citric acid cycle. So this is a single reaction. It takes us from pyruvate to acetyl-CoA. A lot of times people wonder why you don't just call glycolysis all the way up to acetyl-CoA, and the answer is what we talked about at the end of the last lecture, which is that if we go anaerobically, pyruvate does not go to acetyl-CoA, it goes in a different direction in lactic acid in humans. So that's why, but you can kind of consider stage 1 to be glycolysis plus this transition reaction. And then stage 2 starts with acetyl-CoA. So stage 2 is actually going to completely finish breaking down glucose all the way to CO2. But we're not going to have made any ATP yet. Stage 3 is going to be taking all of these electrons that during glycolysis, and we'll see today in the TCA cycle, we're going to pull all these electrons off and put them onto NADH and FADH2. We've already seen one NADH. We saw this in glycolysis. We're going to see a whole bunch more, and we're going to see FADH2. So stage 3 is just recycling these electron carriers back to NAD plus and FAD through something called the respiratory chain or the electron transport chain. And this is where the oxygen comes in. So oxygen is required for this entire path part just because it's required for this. Because if we are unable to recycle NADH and FADH2 through the electron transport chain, we are not going to be able to do the TCA cycle either, because we're not going to have enough of these electron carriers in the oxidized forms to do it. So we're not going to see any oxygen in the citric acid cycle, although I'm going to tell you this only can happen under oxidative conditions, and that's because we can't recycle the carriers. So today we are going to talk about stage 2 and also this transition reaction, and then next time we will talk about stage 3. So first, starting out with that transition reaction, the oxidation of pyruvate. So here let's look at pyruvate. And if we look at, we're going to acetyl-CoA. So this has 3 carbons in it. Acetyl-CoA has 2 carbons in it. The third carbon has been lost as CO2. So this is our first loss of CO2. Right? We didn't get rid of any carbon in glycolysis. We just moved around. We chopped it in half and we moved it around. But we haven't gotten rid of any carbon as CO2. So here we've gotten rid of one of them. And we still have acetyl-CoA, which is a 2-carbon molecule. So what's happening here is we're going to remove the CO2, and then we're going to put on the CoA. So on here, this is called this reaction. Whenever you take so this is an alpha keto acid, which means that we have the acid, and then we have on the next carbon over, we have a ketone. So if you take an alpha keto acid and you convert it to a thioester, this is called oxidative decarboxylation. Oxidative decarboxylation. I think it's helpful to sometimes, right, classify things. You don't necessarily always want to stress about it, but classifying can help you recognize when you see new things, right? Which means this is an oxidative reaction, it's a redox reaction. And we see here's our NAD plus NADH. And it's a decarboxylation because the CO2 leaves, right? This is performed by a complex called the pyruvate dehydrogenase complex. We're not going to focus on the mechanism here. We will come back to this in term 2 and talk about that if you're taking that course. But what we're going to talk, what we care about here is knowing what happens between pyruvate and acetyl-CoA, and knowing that we are going to create an NADH in the process. We also are going to use 3 other cofactors, but these are cofactors that are going to be regenerated within the course of the mechanism, so we're not actually creating anything. You notice there's this FAD, but we're not creating FADH2, so this is not going to have an effect. These are basically still essentially a catalyst. All right, so those are the 2 key parts to that, is sort of what actually happens and the fact that we're going to make an NADH. This has a very negative delta G. This is a very irreversible reaction. You cannot go backwards. So in humans, we can take pyruvate and go to acetyl-CoA. We cannot take acetyl-CoA and go backwards to pyruvate. So just to remind you what CoA looks like, it is a very large complex molecule. It's got this adenosine diphosphate thing that's also got a phosphate on carbon 3, and it's got a long pantothenic acid linking chain, and then at the very end here we've got a reactive thiol. That's the only thing we're really going to care about. So oftentimes your book, and I do this too, we actually draw out the S from the thiol. I just want to be clear that S is part of CoA. So even if I'm going to write S-CoA, that's the same thing as writing just this. So I could just draw it like this, but I'm just trying to remind you that there is a sulfur there that is part of CoA. Right, so of course this is an acetyl group, and so that's what's acetyl-CoA or acetyl-CoA. Either pronunciation, I've heard both pronunciations, and honestly I switch back and forth between them. So, all right, so we're not going to worry about really, none of this changes during the reaction. This is all we care about. All right, so sometimes people wonder, all right, then we have acetyl-CoA. Why don't we just oxidize it again, right? We could oxidize this guy up to carbox CO2, and then we just have that in methane, and we just have to oxidize methane. And the answer is there's no good biochemical way to oxidize methane to CO2. We just don't have the enzymes to do it. So instead, what we're going to do is a rather complicated multi-step pathway where we do not go straight from CoA, but we're going to do multiple steps in order to turn them both into CO2. So this is called the citric acid cycle. Other names for this are the Krebs cycle, named after the person who discovered it or wrote it down, basically. The tricarboxylic acid cycle, or the TCA cycle. So TCA actually stands for tricarboxylic acid cycle, but I mean, I also sort of feel like it could stand for this. So I generally call it the TCA cycle, completely honestly, because it's the shortest. But I may also call it the citric acid cycle too. Generally speaking, I think we're sort of moving away from naming things after people, and we're moving more towards naming things towards what they do. So although you should absolutely be aware if somebody talks about the Krebs cycle that this is what they're talking about, I'm not generally going to use that term. All right, so this is a cycle, and we're going to, it starts up here. So here's acetyl-CoA. And sometimes people get confused because it's a cycle, because it doesn't seem like we're making any more of these things because they just keep looping back around. And that is true. So any of these intermediates, so citrate, cis-aconitate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate, we're not actually making any more of those. That is not the point. You almost can think of this as one really big enzyme reaction. Now, it's going to have many more enzymes, but in the same way that an enzyme has to go back where it started, you almost could think of these of these atoms in the oxaloacetate as being part of an enzyme. They get maybe temporarily attached, but then by the end of the reaction, we're unattached. So what we are doing here is the things that come into and out of the system, the cycle. So acetyl-CoA comes in. It's not an intermediate. And then we also have a few other things coming in. We've got a couple CoAs coming in, but then they're going to go back out, actually. So really, acetyl-CoA is what's going to come in, and then coming out are going to be CO2s. So by the end of the citric acid cycle, so we brought in 2 carbons, and throughout the whole cycle, we see 2 carbons leave as carbon dioxide. So these carbons we've now essentially produced the rest of our CO2. And we also have produced 3 NADHs and 1 FADH2. So if you look at this, FAD should come in and NAD plus should come out. They're not showing that, but all these places where NADH is coming out, NAD plus would be coming in. And this step that makes FADH2, this FAD would be coming in. So those things are coming in, and really the overall gist is acetyl-CoA is going in and CO2s are coming out. However, I want to point out to you at this point that this has some nice color coding here, and the carbons that come in as acetyl-CoA in that round are labeled pink. So you can track them, pink, 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 pink. They are still here in succinyl-CoA, even though both the carbons have been lost. So it is not directly that these are going to be oxidized to CO2. It's that these 2 carbons come in and different carbons get released. But still the overall reaction can be acetyl-CoA goes to 2CO2. All right, so just don't let that confuse you too much. So it's 8 steps. We are going to go through each step individually, but this is a pretty good generic slide to help you once you've seen it all to look at it. All right, so step 1 is formation of citrate. This is essentially the committed step. Once we're in here, we're going to go through the citric acid cycle. There's actually a lot of other things acetyl-CoA can do, but once it's been put onto oxaloacetate to make citrate, we're committed to going through the TCA cycle. So making sense with that, this is an irreversible reaction and done by citrate synthase. Again, I'm not going to ask you to know the names, but some people find it helpful, right? Because we're synthesizing citrate. Okay, so here we have a new molecule, oxaloacetate. It has 4 carbons. So it's basically got 2 carboxylic acids on each end, and then one of the carbons in the middle is a ketone. And what's going to happen is acetyl-CoA is going to sort of get this methyl group. It's going to get joined to this carbon, and this is going to open up into an alcohol. So that may not be immediately obvious how that could happen, so we're just going to talk about the mechanism. So first what's going to happen is oxaloacetate is going to bind deep in the pocket and then acetyl-CoA. So this is going to form a ternary complex, and it's got an ordered mechanism. You can't bring in acetyl-CoA first. This figure just shows you an acetyl-CoA analog, and that's simply so that you can get the crystal structure without it very quickly forming the reaction. An analog, everything will bind where it should, but it won't actually complete the reaction. That's the only reason for that being there. The mechanism is going to go like this, but you can look at the overall reaction as well. So here, let's start here. So we have oxaloacetate bound deep in the pocket, and there's a histidine important here, and another histidine here, and aspartic acid here. So the first thing that's going to happen is the aspartic acid is going to deprotonate the methyl group. Again, kind of unusual. But then these electrons in this bond move instead to make a double bond between the carbons, and the electrons on the second bond of the carbonyl go deprotonate this histidine. Okay, so now down here we have an enol intermediate. So we have this double bond and then the alcohol, so that's an enol. And what's going to happen is we're immediately going to reverse that. The nitrogen is going to take the hydrogen back. We're going to move the electrons back down into the double bond, and now these double bond electrons are going to go and attack the carbonyl. So this is going to be it's going to be a nucleophile because these electrons want to go bind to something. It's going to attack a carbonyl carbon. Now this carbonyl is going to open up, form a tetrahedral intermediate, and take the proton off of this histidine. So now here we have this CoA, and we've got this carbonyl methyl group, and then this bit right here. So then all that's going to happen for the last step is water is going to come in here and replace CoA. So the CoA was only there for a hot second in order for this to work, and now we've lost it again. So now we just have citrate with no CoA on it. I am not going to ask you to draw these compounds by heart, so I'm not going to ask you to draw oxaloacetate or draw citrate. That said, I might give you oxaloacetate and acetyl-CoA and ask you to draw what the product is. So some people find it helpful to memorize what this is. If they know that citrate comes after that, they find that helpful. So I'm just going to say that's up to you, what you find helpful in order to learn it. So you should be able to draw the mechanism in the next product, even if you can't identify that it is citrate. The next step is isomerization to isocitrate. So the difference here is that citrate has basically three carbons, and then each carbon has a carboxylic acid coming off of it, and the middle carbon has an alcohol. Isocitrate, the alcohol is on what we could call the second carbon. So it's just been moved. So that's all we're going to do is we're moving the oxygen. And what actually we're going to do, we're going to dehydrate this to the double bond and then rehydrate it in the opposite direction. So the oxygen doesn't really move. It's a different oxygen that comes in, but we don't care so much about that, right? This has a positive delta G, so we know that when that happens, this means that this is probably a reversible reaction and that we keep the concentration of isocitrate lower so that the Q will be less than 1, and that can pull this delta G into the favorable range. Step 3 is oxidation of isocitrate, and it's going to be oxidation right here. So again, we have this double bond, we have the carboxylic acids off all three of these middle carbons. This one has an alcohol. We want to oxidize this alcohol to a ketone. All right, and we're also going to, in the same enzyme, we're going to lose the middle carboxylic acid. So the oxidation actually happens first, and this is going to be, so it's an oxidation, so we need a redox cofactor. We're going to use NAD+. This is showing you this can sometimes use NADP+. We are going to consider that it uses NAD+ only for purposes of keeping things simple. So NAD+ goes to NADH and H+. And we've seen these things before. Basically what happens is something comes in, takes this proton, collapses to a carbonyl, and then the hydride goes and moves on to NAD+. All right, so hydride transfer. Now we've already made that oxidation, but then we're going to decarboxylate it. So carboxylic acids are actually pretty good at decarboxylating as long as there's some place for these electrons to go, because carboxylic acids already have a C and two O's. We just have to bring this guy down and make the second double bond, and that's pretty good. But these electrons have to be able to go somewhere. And it turns out they couldn't go anywhere as long as it was an alcohol, but once it's a ketone, we can temporarily make a double bond here and open up that ketone we just made into basically an O-. But then it's going to collapse right back down, and the double bond is going to find an H. That H is going to be bound to this carbon because that's the one that needs another bond. So then we have alpha ketoglutarate. So this alpha keto comes again from the fact we have a carboxylic acid here, and then the alpha to that on the next carbon, there's a keto. And then the glutarate may sound a little bit like glutamate to you. So if that does, you can notice that the rest of it looks like the glutamate side chain, CH2, CH2, COO minus. Step 4 is going to be oxidation of the alpha keto glutarate to succinyl-CoA. So we're also going to bring in a new CoA. And I want to point out in this version of the book, they have a horrible typo. This is not what alpha keto glutarate looks like. This is not even like what is even going on here. So just don't look at that. What's on the previous slide in the previous figure is the correct one. So just make sure you're looking at the right thing. So here we've got this. We're just going to start from here. And this is another dehydrogenase complex. So this is actually very similar to the pyruvate dehydrogenase complex. We have an alpha keto acid, and we're going to do an oxidative decarboxylation to lose this CO2 and then replace on that carbon the CoA. So we can see that that's what happened. So this ketone is this ketone right here. We've lost a CO2 and we've replaced the CoA on there. So this is another irreversible reaction. There's not very many irreversible reactions in the citric acid cycle, but this is another one because these dehydrogenase complex really only go one direction. So now we've lost one of our CO2s. And we can look at the similarities here between the step 4 in the citric acid cycle and then that pyruvate dehydrogenase complex. They act on different molecules, but what's going on here is all that matters. We have this alpha keto acid, and it's being converted to a thioester. Right? What's going on here, I mean, it matters. They're different ends. They're different complexes. The complex that does this cannot act in the citric acid cycle, but the mechanism is the same, basically the same. It also creates an NADH in both cases and gets rid of a CO2. We're not going to talk about this one this term, but there is another dehydrogenase complex that is part of the breakdown of isoleucine and similar ones also in leucine and valine that do essentially, again, the same thing. Let's see this going on. And this is likely an example of the way evolution sometimes works, where we duplicate a gene. So one of these probably happened first. To be honest, I don't know which one it would have been. It might have been this one, but I could make an argument for this one too. So one of these probably happened first. It also even could be something totally different, just some other dehydrogenase complex. And the genes got duplicated, and then one of them started specializing in dehydrogenasing pyruvate. The other specialized in dehydrogenating alpha keto glutarate. It's also possible that the initial one actually could work on both of them, but not as efficiently, and when it got duplicated, they could specialize and work much better. So, and maybe even this one too. So oftentimes they look very similar. They're both complexes. They have the same mechanisms. They have very similar enzymes. So that's why we think that that's probably how that happened. Again, I also just think it's helpful so you don't have to learn every new step separately. If we've seen something similar before, it's helpful to be able to classify that. All right, step 5 is the formation of succinate. So succinyl-CoA and succinate should sound very similar to you, so we're going to just lose that CoA again. These CoAs are put on because this is something clearly that evolution figured out how to do, which is do this oxidative decarboxylation that puts a CoA on. But we don't really care about the CoA being there, so usually it's going to leave in the next step. So here's succinyl-CoA, and I just want to point out they've sort of changed the way they're drawing it. So here's 1 carbon, 2 carbons, 3 carbons, 4 carbons. 1, 2, 3, 4, right? So we still have 4 carbons, but they've sort of changed the way they're drawing it. And this one is actually also going to create a GTP. I forgot to mention that, I suppose, in the first round. The other thing that happens in the TCA cycle is we create a GTP. This reaction has a delta G very close to 0, so is reversible. And we're going to talk about the mechanism here because you may not immediately be able to see why we might want to or how we might make a GTP while doing this. So the mechanism, we're going to start here. So here's our succinyl-CoA. And what's going to happen is that the inorganic phosphate is going to come in here and kick off the CoA. So CoA is gone. And now we have this phosphorylated version, but we don't really want that. So what we're going to do then is transfer the phosphate eventually onto GTP. But this enzyme is a ping pong enzyme, and it's not a turn-in complex. So what's actually going to happen is a histidine in the protein is going to take the phosphate, breaking this bond, and releasing succinate. And then we have a phosphorylated histidine, and then GDP can come in, take the histidine, take the phosphate off the histidine, and then we're back where we started. So succinate is actually one of those things that might be useful enough to learn what it looks like. If you know sort of bits along the way, it might help you. Also, succinate I feel like is pretty easy. It's 4 carbons, and the first and the last carbon have CO minuses on them. So this is a symmetrical molecule and pretty simple to know what it looks like. Step 6 is oxidation to fumarate. So this had these middle 2 carbons are methylenes, so they're fully saturated. And fumarate is now, it's like succinate, but you've made the double bond. It's not really an alkene because we've also got these going on, but these carbons are alkene carbons. And this particularly forms the trans. So fumarate is the trans version of this. If it had formed the cis version, it would be called malleate, and it does not form this. So we do have to take hydrogens off in appropriate ways to make that work. And the cofactor that does this is FAD. So generally speaking, if we are taking 2 hydrogens off of what's essentially an alkane to make an alkene, we tend to expect that FAD is going to be the one that does this. If it's also involving an oxygen somehow, usually it's an NADH. So we're creating an FADH2 here, and this is another one of our redox cofactors that we are eventually going to recycle in order to make ATP. And this also has a pretty equilibrium reaction. It's delta G of just 0. So we've made this trans fumarate, another molecule. It's probably useful to know what it looks like. That way you just don't, it's just easier on you. Step 7 is just hydration to malate. So just to be clear, malleate is the cis version here. Malate is when we're going to add water across that double bond. And because we started with fumarate, we exclusively make L-malate. This is a chiral carbon right here, as opposed to what would be called D-malate. That hypothetically, if we would hydrate this, we would get D-malate. But we don't have these things. All right, so this is just hydration, adding water across the double bond. And so this is going to happen like so. The OH attacks here, forms a carbanion for a very brief moment before that carbanion finds a proton and forms malate. So again, delta G pretty close to 0, considered pretty equilibrium and reversible. And then the last step, we have to reform oxaloacetate so the cycle can continue. So here's malate, and malate looks like succinate, but it has an OH on one of the middle carbons. And oxaloacetate looks like this, but it's got the carbonyl there. So what we need to do is oxidize this alcohol to a carbonyl. Usually what we use is NAD+, and this is no exception. And so we're going to oxidize that. All right, and this one looks like it has a very positive delta G and therefore should not be favorable. But again, what happens is that the next reaction, step 1 again, remember, was irreversible. And so oxaloacetate is constantly getting pulled very quickly into that reaction. So we never build up any of the product, and that leads us to a Q very less than 1, and therefore shift the actual delta G to be pretty close to 0. Right? So we've made oxaloacetate back again, so now we can go back through the cycle, but we've also produced all these things along the way. This figure is just a very simple one that doesn't show any of the structures, but it gives you the names, and importantly, you can really see those things that are going to come in, just acetyl-CoA, and then leave CO2, 2 CO2s, and then we form 1, 2, 3 NADHs, 1 FADH2, and 1 GTP. What they're showing you here is that GTP can be converted with about a delta G of 0 to ATP, so we can consider essentially that we made an ATP here. The enzyme happens to put it onto GDP instead of ADP, but we can switch that very easily. So it's essentially like making an ATP. So here's the overall reaction, and this is reasonably important to know, these 3 NADHs and FADH2 and GTP. All right, and now the only thing we have to do is talk about how we take these NADHs and the FADH2 that were made along the way, and we make ATP with them. So that is what we're going to do in the next lecture.